Questions from anything we covered last time we were here? All right, so we talked a little bit about dosage forms, kind of the definitions we're going to be using throughout this course. I want to finish up talking about kind of receptor signaling. How's this actual information being carried from the drug to the receptor to whatever the downstream sort of effect we're expecting to see is going to be. So several types of receptors are available. Again, different targets may be considered receptors, right? You may be targeting antibodies, you may be targeting actual cell surface receptors, you might be going down into the nucleus, actually changing things like gene transcription. So lots of different things can be considered receptors, as we'll find. Now, the body communicates between its own cells in various ways, and we're going to see that in some cases drugs will try to mimic this, and so I'll show you a couple of different ways that can occur. You guys know of any ways that the body transmits signals from one another, sends its own drugs out to communicate to other cells? Yeah, neurotransmitters is a very good one, right? So that's actually happening right at the synapse between, say, two neurons, or say between a neuron and a cell. That could be one good one. Any other ones? So sodium channel may be a target, maybe a type of target going to be hitting there, but in a lot of cases you'll see that's like, along with neuronal transmission and things like that.
Would you consider like when you have an inflammation, the cells are communicating?
Yeah. Sometimes you're seeing that cells will release inflammatory mediators that get released into the bloodstream, right? And they can communicate to other cells, that may be very far apart from one another, in order to recruit those cells to cause an inflammation. Absolutely. A couple ones. Just know that receptor is basically going to be the specific protein that is going to be the binding site for whatever we're talking about, whatever ligand we're talking about. The effector is the mechanism that then gets activated by that receptor. We'll go over countless examples as we go through these courses here. Now, that receptor, when that actual signaling occurs, there's usually going to be a second messenger system that gets activated after that, and that's going to be an important thing where a lot of drugs are going to be working through. So we'll see what those look like in the actual next section. So just a couple of different types. You can have things like contact-dependent cell signaling. This is where cells are going to be in contact with one another and can communicate. You run into problems with this, especially with, usually when cells kind of grow as much as they need to, they start to have this kind of cell contact-dependent inhibition, where basically they realize, hey, I don't need to keep growing anymore. I've taken up all the room I have. Can you think of any disease states where that might go wrong? Cancer is a big one, right? So again, that's one of the things you'll find. There's mutations that lead to, they lose that cell contact inhibition. So they keep growing and growing, despite the fact that they don't really have any room to do so, right? That's when you have those tumors and things like that form. So that's one type. Paracrine signaling is going to be where basically cells are able to communicate with cells that are nearby, but maybe not in direct contact. So for instance, with that inflammation, imagine if you were to have, say, for instance, some inflammation occurring there, maybe some monocytes or something are degranulating and releasing mediators or communicating to cells nearby to try to recruit them for that inflammation. That could be one thing you could see with that. That synaptic signaling, this is going to be very, very important when we're talking about neuro stuff, when we're talking about psychiatric medications, seizure medications. All of that's going to become very important because oftentimes we're either dealing with the connections between two different neurons, or we're dealing with the connection between a neuron to some sort of target tissue. So for instance, when we're dealing with things like SSRIs, like for depression, anyone know what that means, that stands for? Selective serotonin reuptake inhibitor. That's directly affecting this synapse here, where it'll prevent things like serotonin from being reuptaken, so you have more serotonin out in the synapse, right? So that's going to be one of the main mechanisms you're going to see for a drug like that. So we'll talk about those much more in detail when we get to specific examples in Pharm 1 and Pharm 2, as we'll see. And then endocrine signal, this is a very important one, especially for things like, all of your hormones you'll be releasing, whether, and you guys have covered like the HPA axis, the hypothalamic pituitary axis, as far as different endocrine systems go. So how things like, luteinizing hormone and follicle stimulating hormone are going to be, stimulating things like, the sex organ development or menstruation and ovulation, all those sorts of things. That axis is very important for endocrine signaling. Basically, the cells are releasing their signals, their drugs essentially, into the bloodstream, and that can go to all over the body and cause its effects, right? So for instance, the pancreas releases what primary hormone? Insulin's a big one, right? So it can release that into the bloodstream. That can then go along and go and interact with all sorts of tissues to drive, glucose uptake by the cells, do all kinds of different things. So that's a very important cell type of signaling we're going to see, and a lot of drugs are going to be mimicking this, because again, when you have drug being absorbed from the oral route, or if you give a drug IV, it's getting to that systemic circulation, circulating around and getting to the whole body essentially. All right, so that's an important type of cell signaling we'll see here. Okay, so different targets you can see for receptors could be things like enzymes, right? So what do enzymes do? Yeah, they make a reaction happen much faster than it would be otherwise. You may find that you can inhibit enzymes. You may find you can make enzymes work better than they would normally with the effects of a drug. So a good example of this would be something like methotrexate. Anyone ever heard of the drug methotrexate? Used a lot for like rheumatoid arthritis or other types of autoimmune conditions, but cancer is another big one we use it for pretty frequently. And what this actually does is it actually will interrupt the processing of folic acid, essentially, right? So basically, by inhibiting folic acid utilization, you're going to be preventing these cells from making new DNA, essentially, by making thymidine, that nucleotide. If you can't make thymidine, you can't make DNA. Guess what happens to the cell? Dies off, right? So again, that's where you can see that you can help to inhibit the immune system by doing this. You can inhibit cancer cells from growing continuously, and so that's an example of just an enzyme you may inhibit in order to make, to get whatever kind of clinical effects you're looking for, right? And again, I'm not going to have you memorize these exact examples. I'm not going to ask you on a test, hey, what does methotrexate do, right? That's going to be for later classes, right? But I may say, hey, what are potential targets for these medications? You may say, okay, well, I know an enzyme could be a potential target, right? I know, these different cell surface receptors could be a target. Those are the kind of the concepts I want you to get now. You could be affecting transport proteins. So you mentioned sodium channels, and this is actually a really big one. We'll be affecting, especially with things like the neuro drugs. We'll be affecting this, especially with cardiac drugs as well. Usually, transport proteins are used to channel something from the outside of a cell to the inside of a cell, or vice versa, depending on how the case may be. And so, if you inhibit that transport, you can modify how the cells are going to be functioning pretty significantly. So, for instance, we have a drug. Anyone ever heard of digoxin before? Something we use for CHF, more commonly sometimes used for atrial fibrillation in some cases. And so what this actually does is there's a protein called the sodium potassium ATPase pump. People have heard of that before? It's a very important pump. I want you to at least, when you're going into pharma, we're going to talk a lot about action potentials. We'll talk a lot about that sodium potassium pump. But what's its main function? Maintains resting membrane potential, right? So again, every neuron or every cell that's going to be conducting an action potential has a resting membrane potential, right? It's usually negative, and that sodium potassium pump is really important for that. And so digoxin can actually come along and inhibit that pump, and by doing that, you cause some downstream effects that actually cause more calcium to be released within the myocytes. And when you have more calcium being released, if you go back to think about how the muscles function, you need calcium being released to cause actin-myosin to interact with one another to cause a contraction. Same thing for the heart. More calcium means the heart's going to be stronger. I'm going to get better contractility out of the heart. Okay, so that's one example where you could affect an actual transport protein. By inhibiting it, you can make, say, the heart beat stronger than it would otherwise, right? And that's usually a problem with someone with CHF, is they have a hard time with that left ventricular contractility, right? You can affect things like structural proteins. So, for instance, proteins are useful for helping with things like mitotic division or providing cell structure. Anyone ever heard of gout before? Anyone know what gout is? Yeah, a lot of uric acid buildup. You're having that crystallize out, essentially, or precipitate out. Anyone know what usually gets affected, or the kind of the classic thing gets affected? The big toe, the great toe, they say. I think it's just an okay toe, but some people call it the great toe. But you can find that by using things like colchicine, you can actually inhibit those microtubules from actually forming in the first place, and you can prevent things like mitosis from occurring, right? You remember, if you go back to looking at mitosis, when you're actually pulling the chromosomes apart, that's using those microtubules. If you inhibit that, guess what? Cells don't pull apart, they don't undergo mitosis, and then they get signal off for apoptosis, right? That cell death. So that's one case where you may see a drug being used to actually inhibit some structural proteins. And I'm going through this kind of fast, but I just want to make sure we get through all the material before your exam is going to be next week, right? Now, the main drug receptors we're going to be finding are going to be including things like ion channel receptors. These are the primary ones we're going to run into. Gprotein-coupled receptors, enzyme-linked receptors, and then intracellular receptors. We're going to go over these in more detail here in just a minute. So, and again, looking at the difference between, say, something like a cell surface receptor versus cytosolic, or say a nuclear type of receptor, basically it's right there in the name. Cell surface receptors are going to be where the drug interacts right there with the cell surface, right? So the receptor will be on the outside of the cell and can then interact with it, and then it will cause downstream effects afterwards. If you have something that's more of like a nuclear type of receptor, that's where the drug will actually get into the cell, either through passive diffusion or it will get through based on transport proteins, however it gets in, and then it can go down to the nucleus where it can do things like change how proteins are being produced, what genes are being expressed or inhibited. You can cause lots of big changes here with actual cell machinery, essentially, by affecting the nucleus. And so getting into the different types of cell surface receptors, we'll find that the ion link channels, the G protein link channels, and the enzyme linked are all going to be examples of cell surface type receptors. Okay, so the ion link channels are basically going to be also called ligand-gated ion channels or ionotropic receptors. These are basically channels that, when open, will allow some sort of ion to flow through. Some of these can be very specific. For instance, you can find things. You have ever heard of the neurotransmitter GABA? What does GABA do? It's the main inhibitory neurotransmitter, right? So if you think about, if you take like a CNS depressant like alcohol or a Xanax or something like that, that is activating GABA. It's making it work better. It's making everything slow down essentially, right? That allows for chloride to flow through these channels, right? When you open that channel up, that GABA channel up, chloride flows in, and what do you think that does to the cell? Makes it more difficult to have an action potential because it's more negative, right? If you think back to your action potentials, remember you have to hit some sort of critical point where you can have a depolarization. You can inhibit that by causing the cell to become more negative, essentially, right? So that's one example. You can find sodium channels that, when blocking that, you can actually inhibit depolarization in those cases. So lots of different means to do this. Some of them are less specific than others. Some of them allow multiple different types of ions to flow through. Some of them are very specific for one type. But again, when you open these, note that things are going to be flowing passively from an area of high concentration to low concentration, right? So that's really important, for instance, when you're thinking about things like action potentials in the heart. It's good to know in the heart that on the outside of the cell, sodium is in a very high concentration. Potassium tends to be on the very high concentration on the inside of the cell, okay? The thing I always think about with that kind of, it sounds like a silly analogy, but someone described our cells as salt-wrapped bananas. So you get a lot of potassium on the inside, a lot of salt on the outside, essentially. And so that's good because I can allow things to flow along the concentration gradient. So when I need to have an action potential, say for instance I want the heart to contract, I can open up sodium channels and they'll flow along their concentration gradient. It's a passive process, does not require any ATP, does not require any energy for that to occur, right? So that's one example you can see there where things are going to flow along their electrochemical gradient. But you're going to find that lots of different ligands can affect this. Neurotransmitters are a huge one, so things like GABA, acetylcholine, serotonin. We'll talk about those, many different examples of those later on in Farm 1 and Farm 2. And again, you're going to find that these receptors can be made up of different subunits, and so based on where you're at in the tissue, you can find that different subunits will confer different activities, essentially. So for instance, in this one here, say in this tissue, you may have, say, 2 alpha subunits, a beta, a gamma, and a delta. However, if you were to, say, maybe go to a different type of tissue, maybe you have 3 alpha and 2 beta, but it's always that same 5 subunits for that particular receptor. Just know that allows us to have different functions in different tissues, essentially, right? And again, you can find that there's lots of varieties here. So, for example, the way that this is manifested is by the fact that by having different proteins being expressed or different genes being expressed in different amounts will allow for that splicing of the RNA to occur to allow for different subunits to be formed there. So that's why there's a little bit of variability depending on the type of tissue you're dealing with. So anyway, so again, many combinations of subunits can make a functional receptor essentially. So as an example of that, let's look at acetylcholine receptors here. And again, acetylcholine is very important for function of things like the parasympathetic nervous system. It's very important for neuromuscular functioning, right? Your skeletal muscle doesn't work unless you have acetylcholine being transmitted. It's important for part of the sympathetic nervous system. We'll look at that later on for affecting some of the preganglionic neurons. But anywho, what you're going to find is there's different receptor subtypes here. So for instance, we have an NM, which is located on the skeletal tissue, and it tends to be excitatory. It's going to do things like increase the permeability of sodium and potassium, okay? However, on the other case, you can have cases like you have an NN type of receptor. It's found on things like the autonomic ganglia. This is what's going to help your parasympathetic and sympathetic nervous system to work, essentially. And then you can have a CNS type of receptor that's also going to be excitatory. So again, different subunits form these different types of receptors. They all have a different little flavor to them, what they're going to do. However, they all respond to acetylcholine the same way, right? They still all have that ability to bind to acetylcholine identically, okay? So again, I'm not going to have you memorize specific acetylcholine receptors here, but just know that there's lots of varieties there. And based on things like, especially like serotonin receptors, there's a huge number of serotonin receptors. By affecting one of them, I can have an anti-nausea drug. By affecting a different one, I can cause things like, migraine to go away. And so it's important to realize when I talk about different types of receptors, you have some familiarity with that when we talk about them in specific examples. Right? Okay. Again, you're going to find that very important for these to function. So, for instance, like nicotine, is going to bind nicotinic acetylcholine receptors in the CNS. It's actually how it got its name. Essentially, we found what the a ligand was for it, and it was nicotine was the first thing we found. You also have things like curare can bind to acetylcholine receptor. By blocking it, you can actually cause paralysis to occur. It's when you go to surgery and you have a paralytic given to you to loosen up all the muscles. That's basically what they're doing essentially, is blocking the effect of acetylcholine. So again, sometimes we're making these work better, sometimes we're inhibiting them. It all just depends on the clinical situation, what we're actually going for there. Okay. So you can get those different examples, but we'll talk about them more specifically in Farm 1 and Farm 2. Okay, the next set we have are going to be our G-protein coupled receptors. This is a huge, wide variety of different G-protein coupled receptors we're going to find. But basically, the same structure you're going to find for all of these is the fact they have the 7 transmembrane sort of spanning regions here of the protein. The drug is actually going to bind here on the extracellular side of the cell, and then it's going to cause some sort of downstream effect. And when it affects something else, we're going to find that's when the second messengers come into play. And we're going to look at specific examples of those in a little bit. So there's a ton of different ones. And so for instance, just looking at like say mice G proteins, there's 1,000 different ones just for smell alone, right? So there's a wide variety of these. By changing the actual amino acid sequence of those G proteins, you can allow it to bind to all sorts of different things, right? We have a lot of those as well. If you think about like your olfactory nerve and think about the process of olfaction, we have tons of different G proteins to respond to different types of smells, right? So to go back to difference between like olfaction versus gustation, what is gustation? Just taste, right? So, if you think about how many different types of taste buds do you have? We have a lot. You have the actual number are a lot, but how many different tastes can you actually taste with your tongue? It's 5. 5 different tastes. You can sour, sweet, salty, bitter, and umami, right? Umami is that secret 5th flavor there. However, foods just don't taste salty or bitter. You have that sense of smell there, and because you have all these different G proteins that can detect all these different little bits of smell, it helps to enhance your taste, right? So again, when you have like a really stuffy nose and you try to eat something, food just doesn't taste right? Because again, you're losing a lot of that activation there. So lots of different G proteins all over the body can have wide variety of effects here, as we'll see. And again, about half of all known drugs that we have work through these G protein linked receptors. So very wide variety here. Again, basically you're going to find the structure is going to be the same for all of these. It just depends on the actual amino acid sequence on what it's going to bind and then what downstream effects it's going to have. But again, this is where the actual drug binds to, and we'll see whatever downstream effect, whatever second messenger is going to be hitting after this. So basically what's happening here when you have these G proteins being activated, normally what happens is you have the actual receptor protein here, and then you're going to have this kind of inactive G protein complex. Essentially what's happening is once this binds to it, you're going to cause this activation to occur. And GDP, anyone know what GDP stands for? Sounds like gross domestic product. So it's guanosine diphosphate, right? So just like we have adenosine triphosphate and diphosphate, this is a similar type of molecule with guanosine instead of adenosine. So here we have guanosine diphosphate. It's actually going to pick up a phosphate group, right, which is going to pick up some energy. So this is sort of an energy-dependent process, and it's actually will then activate these G proteins here. And then this complex can go along and do whatever else it's going to do, whether it's activating different types of enzymes, whatever downstream effects it's going to have. This is what occurs here, right? Now, do these stay active forever? No, at some point you're going to have the whole complex reset itself, and that's usually going to happen when you have this GTP get hydrolyzed. You lose that phosphate group, and then it's going to go back to the inactive complex, and then you can have another molecule of drug come on and bind and start the whole process over again. Okay, that's the general rule how these G proteins are going to be working. Again, once you have the GTP lose that phosphate group, then you're going to find that it's going to go back to the inactive state, and then it'll kind of come back together and then get ready for another receptor activation, and then you can have that activation again occur. It'll pick up another phosphate group, and the whole thing starts over again. So just to give you some examples of different ones, and again, I'm not going to make you memorize all of these, but just to give you an idea. So for instance, we have things like GS proteins. These are going to be the ones that are responsible for the beta receptors, right? So think about beta 1 on the heart. Beta 1 is very important for causing increased inotropy, increased chronotropy, making the heartbeat stronger and faster. This works by activating something like called adenylyl cyclase. We'll look at these enzymes in a few minutes. But by doing that, you increase a molecule called cyclic AMP, and that causes the heart to beat stronger or faster, okay? You can have similar G proteins, and again, these all look the same. You have G olfactory proteins that are going to be good for odorants, things that are going to be you're going to be detecting from the sense of smell. Neurotransmitters, acetylcholine, all these different varieties. But again, they all function very similar to one another. Okay, they all have that receptor molecule that is going to bind to that protein that causes that GTP to be formed, cause the activation of the G protein complex. I would not memorize this chart. No, this is just for example, right? So again, not a ton of memorization for this class specifically, right? Is when you get into Farm 1 and Farm 2, you have to start memorizing like, hundreds of drugs, right? Not to scare you guys, but there's hundreds of drugs out there, right? Thousands in case. But just know that, again, this is more of a conceptual kind of class. I want you to get these basics down so that way when we talk about things like beta blockers or vasopressors, like you're going to understand, okay, well, these are the type of proteins you're going to be interacting with. This is how they get their downstream effects. This is how I cause changes in blood pressure or heart rate or whatever it happens to be, right? Just for example, though. Now, looking at the enzyme-linked receptors, you're going to find there's lots of different varieties here. The main one I'm going to be focusing on, though, is going to be tyrosine kinase receptors. Right? There's lots of other types that are out there, but this is the one that is going to be most frequently found being involved with medications, as we'll see in just a second. If I show you the picture, what it looks like. But this is really, really important. This is good for sort of different hormonal sort of responses. Insulin is going to be working through these tyrosine kinase receptors. A lot of new cancer drugs are actually working through these type of receptors as well. So very, very important for things like DNA synthesis, cell proliferation, etc. Okay, so I'll show you what these are. Basically, you will find that these proteins are basically kind of have two separate components of it. They're identical to one another, and you basically need two molecules of the drug to come in and bind to it, right? So once you have two molecules, and imagine here we have something like insulin, right? Insulin can come and bind to these two inactive proteins. They're going to come together, and then you notice the tyrosine that's right here, it actually will end up picking up a phosphate group. Right, so it needs actually 6 molecules of ATP to be activated, and once that occurs, right, and it's going to be using that from ATP essentially to take the phosphate group off of that. At that point, then it will have whatever downstream effect it's going to have. So in case of insulin when it gets activated, say for instance like the skeletal muscle, right? What does insulin do for the skeletal muscle? Anyone know? It allows for glucose to actually come into the cell. And the way that that happens is because once these tyrosine kinase receptors are activated, it will cause exocytosis, essentially, of these glucose transporters. It will move them to the cell surface to allow for glucose to come into the cell. Normally, these cells are going to be impermeable to glucose, which is good because you want to be able to regulate that. Once the transporters are moved up to the cell surface, it opens up and allows glucose to come in. Okay, but again, it's working through this initial first messenger where the insulin's binding to the receptors. They can become activated, and then you see a downstream effect. Okay. Again, some of these are really good. The tyrosine kinase receptors are starting to innovate things like certain cancer treatments, right? So, for instance, if you have a certain form of CML or chronic myeloid leukemia, there's an actual specific mutation you have where you express a specific receptor. And by targeting that, you can actually target just the cancer cells and spare all the healthy cells, and actually kind of revolutionize how you treat that particular type of cancer with that mutation. So they tend to be more specific, as you'll find. The last one we'll talk about is going to be the intracellular receptors. These are drugs that have to get into the actual cell before they can go and find some target. So for instance, steroids I always think of kind of being the most common ones you're going to run into, whether it be things like hydrocortisone, whether it's going to be steroids like testosterone or estrogen, progesterone. Again, these are all kind of fit under that steroid sort of category. You're going to find that these are all have to get intracellular to get down to the nucleus to cause changes in how genes are being transcribed, what proteins are being produced, etc. Right? So again, these can have very, very powerful effects. It can take a little bit longer though, just due to the fact they have to change things like gene transcription.
It has to be lipophilic always?
So in a lot of cases, you may find they're lipophilic, where they're able to cross that membrane. So that's a good point there, right? So again, they can either pass through just passive diffusion. In some cases, you're going to find they have specific transporters they're going to be working through. So maybe they will have a transporter that will allow them to go through selectively. So it just depends on the drug. Yeah. Question. And again, looking at how they actually go down to the nucleus itself, it can affect the actual DNA. So maybe, for instance, with say, for instance, you're having like a severe allergy, having an anaphylactic reaction. One of the things we'll give you is steroids, right? We'll give you steroids to try to act as an anti-inflammatory. By doing that, those are getting down to those cells where all the inflammation is occurring, and we'll change it so that you're not producing all those inflammatory chemicals, essentially, right? So that's one of the ways we can actually change gene transcription to try to inhibit that inflammation that comes with, say, a severe allergy or an injury or whatever happens to be. Okay, that's just one example, but there's many, many others that are out there. Okay, so those are the summary of the main types of receptors. Again, the first messenger when we talk about that is basically the molecule coming and binding to one of these guys, and these are the most common ones you're going to run into. But there are other types that are out there. There's enzymes you can affect, antibodies, all kinds of different things that can be potential targets here. All right, so any questions on that? All right, now I'm going to head in.